Hello, I'm Ryan Ranjanathan, and I'm here to talk to you today about the microbiome of water. First, we have to discuss the significance of water and why we care about what is in it. Water is one of the key components of survival for all living things. In fact, 70% of your body is made of water. If that water is contaminated, it could lead to illnesses or even death. In our drinking water, chemical disinfections have occurred and was started in the 1990s, where one of the potential factors to the contamination of water was the environment. My primary research was about the microbiome of water, which was conducted in the Walt Lab at Tufts University. First, we have to understand what is the microbiome. The microbiome is a collection of microbes in an ecosystem, such as your body, for example. Within various locations of your body, there will be different microbes. So the microbes that would be behind your ear would be different from the microbes that would be in your mouth. Your body is predominantly covered with microbes. To visualize how many there are, there's approximately 10 microbes for every somatic cell. The main objective of this experiment was to test whether the primary substance that we take into our body could be harming us due to improper disinfections. The primary technique that was used in this experiment is called 16S RNA sequencing, which is used due to every microorganism containing the 16S gene, which is used to identify these microbes. Some of the major microbes could be harmful if found in drinking water, such as coliform bacteria, Giardia lamblia, cyanobacteria, and E. coli, just to name a few. In this experiment, 10 locations were sampled, from a negative control of river water to a positive control of nuclease-free water. It was hypothesized that the treated water and the filtered water would be clear and free of many microbes. To test, in this experiment, we tested 2 different city tap water and 2 city fountain waters found in each respective high school to see whether students are getting treated water to be able to drink. The methods that were used in this experiment was filtration, which took water and forced it through a syringe, where DNA were to be caught in a filter. Then regular DNA extraction protocol was found by breaking open the cells found in lysis, purifying the DNA found in DNA purification using spin columns and a centrifuge, then quantifying the data from the obtained samples using DNA quantification using the qubit and base analysis. The hierarchical clustering was used to determine whether these similarities within the microorganisms found in similar places, also to see in some cases filtration worked. As normal, river filtered and unfiltered are similar. Pure filtered and City 1 fountain are similar. Nuclease-free and bottled are similar to each other. They primarily are found in the same source, but just one sample was filtered from the other. But in case of nuclease-free and bottled, they are both suspected to be clean. Here we see the species diversity results, which show the number of species identified in each sample of water. Some significant results were obtained by looking at the nuclease-free water, which contains 674 identified species, while the bottled water contained 912 identified species, which these 2 samples are supposedly clean. Using qubit fluorescent machine, the amount of DNA found in each sample was identified, and significant results were found in the nuclease-free water that was supposedly containing no DNA. Another case is the high school fountain water containing relatively moderate amount of DNA, even though it is for consumption of students. The negative control was found to have 242 nanograms per milliliter, which was suspected due to it being untreated and containing a high amount of organisms found. Looking further into the genus level classifications, the nucleus-free and bottled water contain Sphingomonas, which can lead to meningitis if consumed, while cyanobacteria was found in high school fountain water, which could lead to a mild rash or worst case scenario, death. These two types of water are primarily used within students and anyone in general that's drinking water. This can cause many kinds of problems and must be treated immediately. In conclusion, a majority of water samples did not contain harmful bacteria, such as the various filters that were used in this experiment, except for bottled water, nucleus-free water, and high school fountain water. Supposedly, the nuclease-free water was containing no DNA samples, so this could lead to some kind of contamination within the experiment. This could cause the bottled water to also have a contamination in the experiment, which could lead to further experimentations and further trials to see if the same results are obtained. This, in the case of the fountain water, this could also lead to a contamination, but it is highly unlikely due to there was more than just the regular bacteria found in it. Any questions?